Every day we hear about the challenges affecting industry and our broader society. Trobe University, as a leading university, has a critical role to partner and collaborate with industry and the community to help solve those challenges. The industry engagement team enables that linking between the university and industry. Our collaborations aim to take research from the lab bench to the market and into the homes of our community. For business or industry, you can collaborate with universities for a number of reasons. By working with us, our industry partners have access to new ideas, new research, new technologies. They have the ability to partner with our researchers and collaborate on the development of their new products. Connecting with us also provides access to our talented student cohort for developing your future workforce.
Good afternoon, everyone, and welcome to the Frontiers in Infectious Disease Innovation, a featured session in the 2025 La Trobe Industry Innovation Series. To begin with, I'd like to acknowledge that La Trobe University campuses are located on the lands of many traditional custodians in Victoria and New South Wales. Here in Bundoora, we're located on the lands of the Wurundjeri people. We recognise their ongoing connection to the land and waterways and value their unique contribution to the university and wider Australian society. La Trobe University is committed to providing opportunities for Aboriginal and Torres Strait Islander people, both as individuals and communities, through teaching, learning, research and partnership across all our campuses and within our work. Hello, I'm Professor Ashley Franks. I'm the co-leader of the Lab for Applied Environmental Microbiology here at La Trobe, and I also serve as the co-director of the Mallee Regional Innovation Centre. As your host this evening, it's a pleasure to welcome you to our campus, specifically to the Digital Innovation Hub, located with our new University Innovation Centre, the Eagleworks, this whole building that we're sitting here within us. Making advancements in infectious disease is a worldwide challenge. The COVID-19 pandemic highlighted our global vulnerability, and it also demonstrates that new innovations and cures are within our reach. Tonight we'll explore this critical topic and bring together the leading voices from Moderna, CARB-X, Deftbiotech and BioBreak Therapeutics. Key discussion points will include advancing innovation with mRNA medicines, accelerating the development of novel products to tackle antimicrobial resistance, Deftbiotech's breaking through antifungal peptide, and harnessing the power of antibiotic technology. Global impact has to start somewhere, so we're making this happen here, right and now. So I'm glad to see so many faces here this evening. We're a passionate community of industry professionals, researchers, academics and graduates who are on the ground transforming lives every day. As we hear from our incredible speakers, I encourage you to think about the connections between their work and the broader ecosystems that drive progress in infectious disease and innovation. If discussions inspire new ideas or spark your curiosity, please don't hesitate to reach out to me directly or speak with our Darcel or Bruce from the industry engagement team. I'm going to make Darcel and Bruce wave because I've got the power to talk about our collaborative operations, and they're always here to help and always discuss and talk to you about further ideas and further opportunities and how the university can help. And on that note, let's dive into the program. This event includes a 1-hour session of presentations from leading experts across industry and academia. You'll be hearing from them shortly, but before we begin, I'd like to take a moment to thank Dr. Richard Arm, Dr. Felicia Pradera, Emeritus Professor Marilyn and Professor Begonia Harris for joining us and generously sharing their insights and expertise to give up this time so that we all can be entertained by the work. Following the talks, you'll have the opportunity to engage directly with our speakers during an audience Q&A session and connect with fellow professionals during the network event after the segment with some more drinks. And on one last point, please note that there'll be filming and photography will take place during the event to capture its highlights and atmosphere. Every effort will be made to ensure attendees feel comfortable and respected throughout. So with that, I'd like to introduce our first speaker. Richard Arm Richard serves as the Chief Scientist at CARB-X, a global non-profit organisation that supports a diverse, innovative pipeline of traditional and non-traditional products to prevent, diagnose and treat infections caused by drug-resistant bacterial pathogens. Richard Arms joined CARB-X in 2019, following almost 20 years in large pharma infection R&D teams, followed by 4 years at a small antibacterial biotech company. Please join me in welcoming Richard.
Thanks, Ash. I hope everyone can hear me. With my screen on full screen, I can't see you, so I can't see if you're nodding or falling asleep. But I'd like to thank the Monash team, the La Trobe team, for putting this product on. Apologies, it is 2:30 in the morning. And I will do my best to introduce CARB-X and some of the work that we've been doing to move products to try and tackle AMR. This is the general disclaimer. The other disclaimer is it is 2:30 in the morning, and so if I say anything wrong, like I just did, please excuse me. Look, bacterial infections, I think we all know the problem that they cause. They cause about 7.7 million deaths per year globally, and 2/3 of those are actually associated with antimicrobial resistance. And the bugs are resistant to drugs that are currently used to try and treat those infections. 5 bacterial species account for over half of these deaths, and you can see from the map at the bottom that the rates of mortality are extremely unbalanced, and we see most deaths in Southeast Asia and Sub-Saharan Africa. I like to think of antibacterials as the foundation of modern medicine. I think we as a society have taken them for granted, and now that's starting to run out, and I think society is at the precipice of suffering the consequence of taking them for granted. Almost 10 million people a year globally undergo chemotherapy, and we've seen recent data coming out from cancer groups showing that cancer patients are more susceptible to infections to drug-resistant bacteria, and infection now is the second leading cause of death for people with cancer. Obviously, transplants, things like dialysis, rely on antibiotics actually underpinning some of the advances that we've made in those areas. 1 in 5 people globally die of sepsis, and so many of all of the sepsis deaths start from an infection, and a large proportion of these are bacterial infections. It is wrong for me to ignore the impact of fungal infections here. In fact, the global burden of fungal mortality and morbidity is actually, from some reports, thought to be higher than bacterial. And obviously, surgery, cesarean sections are becoming more common, especially in sub-Saharan Africa now, and surgical site infections are a leading cause of death, and doctors rely on antibiotics. So without these antibiotics, we risk going back to the dark ages where small cuts and then an infection can actually cause death. And really the AMR paradox is because humans use and misuse antibiotics. On the left-hand side of this slide, you can see clear evidence that the more you use drugs, the more resistance emerges. Antibiotics are one of the few classes of drugs that the more you use them, the less effective they do become because of this emerging resistance. And there's been a lot of epidemiology data done to show that the more drug use increases the level of resistance that circulates within the epidemiological clones in either hospitals or the communities. On the right, I've got some pictures to try and show how we don't value antibiotics enough and we abuse or misuse the antibiotics. Obviously, Australia does very well in terms of agriculture and the amount of antibiotics that are used in agriculture. The US is at the bottom of this chart, doesn't do as well. That's where I'm sitting now, and I'm hoping that the US can actually improve and use less antibiotics in farming and agriculture. At the bottom, you see low and middle income countries. Often you can buy antibiotics over the counter without prescriptions from kids that are running their parents' store, and these antibiotics are then subject to counterfeit they're not necessarily at the right dose. People don't take the full prescription, and that just drives more resistance. And then the billboard that you see there is actually taken in Massachusetts, and I use this as an example to show how much we actually value antibiotics, even in a high-income country like the US. Here is the opening of a new supermarket. They give away free antibiotics while you shop. So even though once you've got a prescription or a teleprescription, you don't even have to pay for the antibiotics. They give them to you for free. And that doesn't sound like something that we don't value them enough. And that's a sad state, I think, on where we are as a society, that these life-saving drugs are just taken for granted. So the AMR crisis really comes in 4 parts. On the top left is the insufficient amount of new drug classes that are being developed. I think we've all seen those slides. The top right is the lack of investment in this space to drive R&D, and I think any academic feels that pinch from national funding agencies as well as investment from venture capital firms. Bottom left is access. The approved agents that are being discovered are not obtaining commercialization broadly across the world. Obviously, this is an increasing challenge in low and middle income countries, but you can see even in high income countries at the top there, like Canada and Australia, this is the last drugs that have been approved in the EU or the US. This shows which countries they are now available in, and I think Australia is batting at about 3 of the last 24. And so access to the right drug to try and treat the patient appropriately is key. And then lastly, on the bottom right is, with all the exits, especially with big pharma, we are losing the workforce of people that know how to discover drugs, and this is something that we'll never be able to get back. And so it's important that we still maintain research in this area to not lose the traction that we have made over the last 15 to 20 years by losing the people that know how to do it. So in response to these crises, CARB-X is a global non-profit partnership that was formed out of the US government and Wellcome Trust in 2016. We have now expanded to be funded by 6 governments and 3 philanthropic organisations. And they give CARB-X, which is housed at Boston University, they give money to us to distribute globally to support the development of novel products. But I will say that CARB-X now is supporting a very fragile ecosystem. This is data taken from our inception in 2016. Since that time, we've had 1,733 applications, and almost 1 in 2 of those applications come from companies or groups that have got less than 10 employees. That's a really small group, and you'll see the bottom bullet point there is these small groups carry risk around the lack of breadth of discipline expertise needed to take drugs and products all the way from early discovery through to clinical development. Sadly, we also see significant attrition. Most of these groups that apply to us have a single asset or very small portfolios, and so everything relies on that single project. And if you look back, I went back to the 2016 to 2019 funding rounds and looked at those companies that had applied to us in those 3 years, and over a third of them now no longer exist. Now, whether they have ceased to exist because their science wasn't good or because they just couldn't get funding, that's hard to tell. But the point is that there's a huge turnover in the ecosystem, and that doesn't bode well for the novel innovation that is needed to bring these products to patients. So CARB-X operates at a global scale. Our goal is to accelerate research and development translationally into human clinical trials. We fund from late hit to lead through to the end of phase 1 or first in human studies. We do support the development of therapeutics, preventatives and diagnostics to treat, manage and prevent bacterial infections caused by pathogens on the WHO priority pathogens list. We select our projects through open public funding calls, and we focus on the performance characteristics of the products and the pathogens that have the largest morbidity and mortality globally. Because of the access concerns that I mentioned earlier, we do require stewardship and access obligations for groups that give our funding, because we provide non-dilutive funding. But we also, and I've got a slide on this in a minute, provide a different type of support model for these often very small companies that need it. So you can see at the bottom there, from the 1,733 applications, we've funded 117 projects. We've deployed just over $500 million US. Those projects have come from 14 countries. We currently have 42 in our portfolio. And importantly, we've actually been able to move 17 products into phase 1 in both the preventative and the therapeutic space, and 5 diagnostic products into late stage clinical assessment. 10 of our projects have had advanced development partnerships. We feel this is important because graduating a project out of the CARB-X portfolio at the end of phase 1 doesn't actually mean it gets to patients if there's no one there to pick it up and help support it through to registration. And so over 10 of our products now have got advanced development partnerships, which is really important to drive these products closer to patients, and we've had 3 that are on the market. So just a brief snapshot of the 42 projects currently in our portfolio. In the therapeutics portfolio, we have a lot of non-traditional projects, proteins, peptides, antivirulants, a lot of this innovation that is coming largely from academic groups. Currently, we have 3 therapeutics that are post-IND and being evaluated in humans. In our preventative portfolio, we have 10 vaccines that cover 6 different pathogens, but we also invest in novel technologies like CRISPR-engineered bacteriophage, microbiome modifying consortia of bacterial strains, antibodies, and also a slow-release formulated peptide. And in the diagnostic space there, we have 5 looking at bloodstream infections, including 4 direct from whole blood to try and speed up that clinical decision-making. We have 4 for sexually transmitted infections, and 3 looking at novel specimens for detection of lower respiratory tract infections. These are the projects that have moved under CARB-X support through the clinic and into the clinic. So you can see we have 6 there in phase 1 clinical trials. We have another 6 that have actually gone straight into or done phase 1B clinical trials. Depending on the indication, we will support studies directly into patients. So if we're looking at something, say, for prosthetic joint infections, the first in human does actually go into patients. So we have 6 in that category. We have 1 that we've supported that has now progressed on with additional BARDA support into phase 3. That's the Vedanta product. We have 2 diagnostic products looking at either rapid detection of bacterial infections in the lung or in the bloodstream that are in final verification and validation studies clinically. And then we have 3 marketed products over on the right-hand side. So I talked about our support model. We like to think that we are more than just giving away money. At the center of our support model, obviously, is a group of experience, both in small biotech and also large pharma, a group of experts that build a company support team in conjunction with our large network of consultants to help those small companies have access to the discipline expertise they need without necessarily paying to have someone on staff. We also run clinical advisory boards specifically for each company as they need them to try and help them with their clinical development plans. I think the biggest differentiator in the success or failure of a small company is actually how well executed their clinical development plan is. Preclinical development is hard, but the clinical development and picking that right patient population to show that differentiated profile of your product is key. And so we run clinical advisory boards with key disciplines and clinicians on there just to give advice of ways to think about how to set up your clinical trials. We run portfolio acceleration tools, which are scientific theses and experiments that will support multiple companies to help answer questions that many companies are facing. So if many companies in our portfolio are facing the same question, we will run experiments on our dollar and then provide the data back to all of the companies in the portfolio. And if appropriate, and this happened last year, we will publish that data so that the whole ecosystem, whether you're in the portfolio or not, will benefit from that. We have expedited access to the NIAID preclinical services when the US government is actually functional. Our companies can get access to those. We have a collaboration with CHI, the Clinton Health Access Initiative, that helps us understand, especially in low and middle income countries, what product characteristics would be most welcomed by different ministries of health in these countries, so that companies get an idea of what they need to work on to actually be able to make that impact into the areas and the countries that need the product the most. And lastly, we have a business development council or an investment day, where, as I said, as our projects are nearing graduation out of the CARB-X portfolio, we have investor days where we bring together them to pitch to a group of advanced developers and VC investors so that they can hear about our portfolio and make decisions about who they want to invest in. So in terms of our funding rounds, the way we work now is we have multiple research themes that are announced simultaneously. They used to be all comers, so anything to do with antibacterial infections we would take. Now they are prioritised to try and balance our investment portfolio in line with our funders' interests. And obviously you can imagine with 10 different funders, there's often quite a balancing act you need to do. The scope of the funding rounds and the budget are approved by our CARB-X funders and then given to us to delegate and execute. And every 2 years we have a strategic review. The next one's coming up in 2026, where we survey large numbers of clinicians and epidemiologists and decision makers in the infectious disease space from around the world to say what are the gaps that are needed. And so that's what helps drive our scope of funding rounds. Typically, we have 2 separate intake dates with the same funding themes. Sometimes we will expand it on the second one, but we'll always keep whatever was in the first one in the second one. And this is to enable groups to generate the data to meet our entry criteria, which is listed on our website. Importantly, the second call for funding in 2025, the round opens on December the 1st. So if you're interested, that's the website. Go there and understand what our funding themes are. You can drop me questions if you like before December the 1st. Once December the 1st starts, I'm not allowed to talk to people, but if you have questions, but before December the 1st, my email is on the last slide. And I'm often asked what makes a successful application. So in three vignettes, this is them. It's never too early to think how your product will be used in the patient journey. And so build a target product profile that will actually bring meaningful differentiation to clinicians that are treating patients, and then incorporate experiments that will emphasize or demonstrate those key product changes. It's so important, as I said earlier, to understand how you will perform your clinical development, how you will recruit your patients, how you will stratify your patients to demonstrate that clinical benefit. And so it's never too early to start thinking about that. Data in your applications. Too often, key data is missing or not included, and therefore it's impossible to assess. This is particularly crucial in a non-confidential 1,000-word expression of interest phase. I tell people that if you have an MIC against Pseudomonas, that is not intellectual property. The structure of the compound is the intellectual property, but tell us that you've got the MIC, don't just say, oh, it's active. And so include as much non-confidential data you can in your expression of interest to make sure you pass that scope check. And lastly, this is a quote from one of our reviewers, that extraordinary claims require extraordinary evidence. And so often we have people come and make these bold claims without the underlying support. We know how hard drug discovery is. We've spent our careers doing it, and we don't mind risk, and we want to understand where the warts are in your program. And so we encourage groups to be scientifically critical and transparent and realistic about where their limitations and where their challenges are in their program, and then embrace CARB-X as a collaborator to help solve those problems, and think of us as more than just a bank that gives money out, because our goal is to help you be successful and deliver a product to the patients that need it. So with that, I will acknowledge all the companies that we have supported. Some of them obviously no longer exist, some of them are no longer in our portfolio, but I will say that we learned something from all of them. They've all been in this space, which is a very tough space, and so I always like to thank all of the companies that we have supported at one time or another during our journey. These are the people that work at CARB-X, an extremely dedicated and extremely passionate group of people in this space, and these are my coordinates if you want to drop me a question if I fall asleep before the panel. But thank you to the Trobe people for putting this on and giving me the opportunity. I would love to be there in Victoria at the moment. I'm sure it's warmer than it is here in Boston, but hopefully my next visit to Australia, I can come and visit you in person. So thank you.
Thank you, Richard, and that's great. And from my own research, I'm glad to see the microbiome engineering has made an appearance there, because I personally think it's an important one, but it was very insightful, and we look forward to hearing more from you during our panel session later on. Now I'd like to welcome our next speaker, Emeritus Professor Marilyn Anderson. Professor Anderson is internationally recognised for her pioneering work in the clinical application of antimicrobial peptides, particularly plant defensins and their role in combating fungal pathogens. As a founding scientist at both Hexima and Deftbiotech, Professor Anderson has played a pivotal role in translating scientific discovery into commercial innovation. Please welcome Marilyn.
Well, thank you, Ashley. So I'm going to change and not talk about bacteria. I'm going to talk about fungal diseases. And Richard, thankfully, told you that they're also important. Our new company, 2 years old now, is called Deftbiotech. And our mission is to commercialise a novel and effective treatment for topical fungal diseases. Why fungal diseases? Well, Richard already outlined that fungal diseases are also a really large problem, not only in humans, also animals and plants. The estimates of the incidence of fungal disease vary, but it's up to 3% of the population of every country. For the US alone, medical costs are estimated to be up to $7.5 billion annually, and that doesn't include treatments for fungal toenail disease, which I'm going to talk about. And like the antibiotics used for bacteria, there's a problem because the widespread use of chemical treatments to control fungal disease in agriculture has led to an alarming emergence of multi-drug resistant fungal pathogens. And it's interesting that many of the pathogens in agriculture are also human pathogens. So we often find in the clinic that people have infections with fungi that are resistant to all known antifungal drugs. So there is an immediate need for new treatments for fungal diseases with different mechanisms of action. So we are working, as Ashley said, with plant defensins. Hopefully I can use this. Here's a structure of a plant defensin. They're just plant proteins that they're naturally occurring. They've evolved with plants. They've been around probably about 400 million years, and they're exquisitely adapted for control of fungal diseases, which affect plants more than bacterial diseases. We've been working on defensins for a very, very long time, so we've got a detailed knowledge of how it works, how it kills fungal pathogens, and one of the benefits of these peptide drugs over current antifungal drugs used in the clinic is that unlike current drugs, it doesn't just slow down the growth of bacteria, of fungi, it actually kills them very rapidly. And we don't have time to go through all of them, but it's active against a very wide range of fungal pathogens. And that's very important because providing protection against one is not very helpful if you're susceptible to infection by a second fungal disease. It's got a unique mechanism of action, as I said. So this molecule recognises and binds specifically to the polysaccharides which are in the cell wall of the fungus. These are yeast cells here, or Candida species. So the defensin here is labelled green. Once it binds to the cell wall, it can enter the cells, and these red areas here are the vacuoles, and the defensin interferes with the structure and the function of the vacuoles. Ultimately, the cells start to leak, and the blue here just shows that the cells of the cell wall and the membrane has broken. I'll continue on here because I'm a bit aware of time. This is just a quick movie. So focus on the bottom two cells here. The outside of the cell is labelled with our green defensin. Here's the vacuole in the middle. You can see the defensin entering, it's the green thing. The vacuoles are all changing shape, and here it is, the cell's dead, all within about 10 minutes. We've decided for our company to go for a market that is large, and the largest market for fungal infections are fungal infections of toenails. It's an infectious disease. People often don't realise that when they have infected toenails. And one of the interesting things is that the current therapies are very ineffective, or they're even toxic. It's particularly bad for people who are immunocompromised or for diabetics. And we've already done a market survey, and clinicians confirm that there is a desperate need for an effective treatment of onychomycosis, and that they would use it if we found one. So our solution, as I've said, is this peptide called pezadeftide. We've already done a lot of work, as I've said. So it reaches the site of infection better than the competitors. It rapidly kills a wide range of fungi that infect nails. It has a unique mechanism of action, which I briefly described. We have a strong patent position. Interestingly, we haven't been able to create drug resistance in the lab, even as we've been trying to force it. And along conditions where we find the other commercial antifungals, you get rapid resistance development. We've already done phase 1 and phase 2 trials, and in both sets of trials it was safe. There were no adverse effects. The formulation is, we've got the formulation solved. It's relatively easy, largely because our molecule is very water soluble, has a long shelf life at room temperature, and we can actually make it. So it's a peptide. One of the things that often limits the development of peptides as drugs is they are expensive to make, but we can make ours in yeast, and we've been doing it reproducibly now in 3,000 litre fermenters. Here's the fermenters. Under GMP conditions, here is our one-step purification process. So we have a reasonable cost of goods that makes us competitive with other drugs. Now let's go back to the market for onychomycosis. It's, again, the numbers vary because not everyone is diagnosed, but it's known to affect at least more than 500 million people globally, but probably 1 in every 10 people on Earth. The market is predicted to grow, currently at about 5.7 billion per annum, and is expected to grow to 9.5 in the next 7 years or so. So there's a huge market for a more effective and affordable treatment than the current topical treatments. So the best topical treatment now for onychomycosis at best only offers a 17% cure, and the oral treatments at best 38% cure. But many people can't take it because it's toxic to the kidneys and to the liver, and you have to have careful monitoring to make sure it's safe. So consequently, only about a small proportion of people actually get treated for this disease. Only about 3 million of the 23 to 40 million people in the USA undergo treatment. Because they know it's very expensive, up to $8,000 per year for the current best topical drug, and this drug is ineffective as well. So what's the story about Deftbiotech? So Deftbiotech has evolved. It's a phoenix that has arisen from the ashes of Hexima. So Hexima was a company embedded here at Trobe University, actually for over 20 years, but the last 10 years was spent on studying antifungal molecules. Hexima completed a successful phase 1 and phase 2A trial. It looked really promising in that trial, but then we had a phase 2B trial, which unfortunately coincided with the COVID epidemic, where we weren't able to give the appropriate monitoring of patients. So we had inconclusive results. Some patients looked great, some didn't seem to improve at all, but we didn't know whether they were consistently taking the drug. So Deftbiotech was established in 2023 after Hexima decided to terminate this project, and we're in the fortunate position that we could accept all the IP from Hexima, and we were convinced that the technology was sound. So the IP includes all patents, trade secrets, formulation, manufacture, all standard operating procedure, data, protocols, etc., toxicology testing, and most importantly, the successful IND application that we had with the FDA. So successful IND normally means that you're free to go ahead with a phase 3 trial, but what Hexima decided not to go ahead with phase 3. Deftbiotech, when we set up the company, we re-examined all the data, and we feel very confident that we have improved the technology. Can't show you in 10 minutes. And we'd like to do another phase 2 clinical trial to test our new dosing and management regime. But here's just some photos of what we have seen, improvements of. So these are 5 separate patients, and you can see rather dramatic appearance improvement within a few weeks, 20 to 33 weeks. The current standard treatment is at least 1 year, so we think that we may get improvement much faster than the current drugs. Got a strong IP position. I realise that we're short of time here, so I'm going to hurry up. Competition. So there are 2 types of treatments that I talked about, topical and oral. Of the oral treatments, really only this one called Lamisil now, which is based on a compound called terbinafine, is available in Australia. It's only available on prescription. This is the one that looks very reasonably good, but this is after taking the drugs which are quite toxic. Here are some topical drugs. Many of them are not available in Australia. Here are 2 that are available in Australia, but you can see that the cure rate is very low, 6 to 9% or 0 to 2.9%. For our drug to be successful, we need to have a cure rate better than 18%, which we feel confident we can achieve. This is just another thing of market value for this drug. USA, 1.9 billion, Japan, 238 million. So you can see it's a large market. What do we want to do? We're trying to raise $1.5 million so that we can do this phase 2 trial and show that we can get really good cure rates. And if we get those results, that will be put us in an inflection point to continue with commercial development and get a partner to go with us to phase 3 trials. Our team, I don't know why Janice is coloured red. Something happened during transition, but you will see this is an unusual company. It's rather female dominant. Really, we only have 1 employee at the moment, and that's Kathy, who's here in the audience, And Adrian and I are here. We volunteer our services, and as do our other friends, Janice Kimball, International Licensing, Susanna, IP Specialist. So we have a team of dedicated people, and we certainly will employ some men when we get some money. I think that's all about it. We don't intend only to deal with toenail infections. The reason we're doing this early is to try and get some cash flow, but we're also developing this drug for other topical fungal infections. The market is not as big for that, so when you're a company, you need to go for the biggest market first. Thank you, Ashley.
Thank you, Marilyn. It was wonderful to hear about the success of the company and the continued work that's going on. Our next speaker is Dr. Felicia Pradera. Dr. Pradera is Director of Moderna's Regional Research Centre and Strategic Alliance in Melbourne, leading R&D collaboration strategies across Australia and Indo-Pacific. With 20 years' experience in research, policy and product development, she has advised national and international governments on the health, security and medical countermeasures, including briefing the US National Security Council on emerging biothreats. So please welcome Felicia.
Okay. So, good evening, everyone. Is that me? Hopefully not. Oops. I'm Felicia Pradera from Moderna. And so a lot of you will be familiar with Moderna. We became a household name during COVID-19. And I really just sort of want to give you an overview of what we're doing here in Australia and how we're growing. And the first thing I will sort of start by saying is a big acknowledgement to mRNA Victoria and, of course, the Commonwealth Government, who worked really hard to attract Moderna, and we're here to really grow and boost the ecosystem. So I'll start really with our vision, and we were established really with that goal of utilising mRNA to treat and prevent disease. So the full name of the Regional Research Centre is for Respiratory Medicine and Tropical Diseases. It's quite a mouthful, but that's a real focus, particularly here in Australia where we sit in the Indo-Pacific region. And our mission is really to deliver that greatest possible impact to people through mRNA medicines. And what makes us different to other companies that work within mRNA and mRNA technologies is that we're solely focused on that platform capability. So for some of you, you might think we're an sort of overnight sensation, but really the company was founded in 2010 and a lot of work was undertaken prior to, of course, the COVID-19 pandemic. And it was in sort of 2019 where things really started to change. Within 67 days of getting the sequence, we had an asset that was ready to start preclinical, and within 12 months we'd gone through the clinical trials to enable a product to come to market, which is quite remarkable for this technology. But what it means by having a platform technology is there is a possibility for that to be adapted not only, of course, to viral pathogens, but to be utilised in oncology, rare diseases, latent vaccines, and other sort of diseases which need to be treated. So when we talk about it, I think most of you in the room, if you've got a science background, will sort of understand the processes here. But effectively, when we're developing assets, the mRNA enables either for the generation of intracellular proteins, transmembrane proteins, or those that are secreted. And depending on the target, of course, it's really important to then protect that mRNA in an LNP for use as a vaccine or therapeutic and as a medicine. So when we sort of look at the way that we constitute this platform technology, it's really highly adaptable, as I mentioned, with lots of applications across infectious diseases. And today I'll talk really about that viral component, because we've had a great talk from Richard around bacterial and of course just now on fungal. But we can tailor that particular sort of target and insert that particular sort of coding region into the platform to enable us to target those emerging and priority pathogens. So when we look at mRNA and its approach for fighting infectious diseases, it enables us to have a rapid turnaround to begin the sort of process, but then also the way that we do our manufacturing and clinical trials. We're able to sort of quickly switch between pathogens. I think what's really quite remarkable for the work that we've done here in Australia, so 3 years ago we broke ground over at Monash at the Clayton site right next to the synchrotron. It was greenfields. Within 3 years, so within 2 years the building was constructed, and this year we received our GMP licences for drug substance and drug product, which means that we have a sovereign capability in this country to address the issues and the threats that may face us in the future. It's quite remarkable, and the team over there has worked exceedingly hard to achieve the goals that they have. I think the other thing from a health security and capability perspective, and we talk about sovereign resilience, but we can't do it by ourselves. And Moderna had really strategically thought about across the globe where it made most sense to develop a manufacturing capability, but also where R&D could be utilised across the globe to support our broader objectives. And so what has happened is we've created what we call the Resilience Network, made up of our counterparts from Australia, the UK, Canada, and of course with Global, which is based in the United States. So we have a really ambitious portfolio, and the plan is really 10 registrations in 3 years. So it's quite sort of an extensive portfolio, and we're tracking very well to this. So as you can see in the red, we of course have our respiratory antigens, so SARS-CoV-2, RSV, and of course our flu and flu combo are in the pipeline. We have both CMV, so cytomegalovirus, and norovirus. We have 2 rare diseases, so propionic acidemia and methylmalonic acidemia. And in our cancer portfolio is our individualised neoantigen therapy, which is we're working with Merck on that. So it's a rather broad portfolio and very ambitious, but for Moderna itself, we have 44 assets in development across a broad range. So these are our priorities at the moment. But I'll just sort of head across and I'll show you a little bit around sort of our priority pathogen pipeline. Whilst the infectious diseases are a little bit earlier in some of them, a little bit further along in the other side of things, there is quite a sort of broad remit that we have and that we're looking at. And all of this work is done in conjunction with collaborators across the globe. So of course SARS-CoV-2 is there, but you'll have heard of Ebola and Marburg and Zika. So we're working really sort of very actively in this space and developing what could be the next generation of vaccinations against these infectious diseases targets. So whilst currently our main focus, as you've seen from our portfolio, is in that respiratory space, and we're seeing an uplift, of course, in our oncology portfolio, Moderna still has a commitment to global public health, and that was one of the sort of foundational reasons that Moderna was stood up in the first place. And so the way that we're doing that is developing a preparedness strategy where we have regional manufacturing. We identify those priority pathogens. And then a program that I really want to talk about is mRNA access. And this is a program that we're doing collaboratively with academics across the globe. And so effectively it enables labs to gain access to mRNA and mRNA LNP, which we provide for free, to undertake work in any of those priority pathogens that I've listed. So This is just sort of an overview really of some of the global partners that have been involved in this program. And what we look at is those promising programs that are really sort of leverage that early development engine to accelerate vaccine development to the clinic. We can't do it all by ourselves. We are 4,500 people globally. Within Australia, we actually don't have R&D positions. We work with academia, we work with other organisations to meet those needs and gaps that we have in our development pipeline, and to identify really new, innovative, creative researchers that can become part of our innovation programs. So, what this access and the mRNA access program gives people the ability to utilise if they sort of join up is we have our unique mRNA design studio. And so we work with those teams on the particular pathogen of interest and working out, what is the right mRNA fragment that they're interested in. We have, of course, the high throughput production, which we do primarily in Norwood for that sort of research grade capability. And then we do sort of this iterative rounds, so people can come back multiple times. They can come for modifications of their sequence of interest that could be another pathogen of interest. But when we take a look at the massive number of pathogens that you are able to take a look at as part of this program, what I will say is that those that are in bold are actually those where we already have programs actively being undertaken. But if you're doing work in any of those bolded pathogens of interest, it doesn't mean that we won't do another sort of a project. We just want to make sure that it is a separate or a different target associated with that particular pathogen. So to broaden really the potential to find a sort of potential vaccine. And this really just gives you an understanding of where that research has gone. So, we really have a sort of comprehensive approach combining that internal R&D, global partnerships and open access to our platform. And this really positions us to respond rapidly and effectively to the infectious diseases of the challenges of today and the unknowns of tomorrow. While not all candidates are advanced to the clinic or registration, the purpose of this research is really to develop and characterise them in those early stage so that they can quickly be activated if the proverbial was to hit the fan. And so, I think that being prepared is a critical part of what we're trying to achieve here in Australia, but also to bring the best innovations and the best possible medicines in our respiratory and oncology portfolio for the public. So with that, I'll say thank you.
Thank you, Felicia. That was wonderful to hear and shows the importance of having a sovereign capability that's globally linked. And our final speaker tonight represents what we are building at La Trobe here at the moment with some of the new things that are coming through. And we'll be hearing from the innovation company BioBreak Therapeutics, led by the CEO, Professor Begonia Harris. BioBreak is on a bold mission to harness the power of antibody technology to tackle one of medicine's most stubborn challenges, chronic and recurrent urinary tract infections. With the global market estimated at $20 billion, their work is not only timely, but it's actually transformative. Please join me in welcoming Begonia for her talk.
Yes, I have 3 minutes to tell you about BioBreak Therapeutics, which is our new venture which focuses on developing novel therapeutics for recurrent and chronic urinary tract infections, addressing a global market valued at $19 billion US. UTIs, or urinary tract infections, are the third most common bacterial infections in humans. There are over 400 million cases globally per year, and these infections disproportionately affect women and the elderly. Uropathogenic E. coli is the leading cause of these infections. This pathogen come from biofilms. These are bacterial structures that shield bacteria from antibiotics and the immune system, leading to chronic infections and recurrent infections. It is estimated that between 20 and 30% of UTIs will become recurrent or chronic. And currently, we don't have effective treatments for these infections. In the lab, we investigated antigen 43, a surface protein on uropathogenic E. coli that basically promotes bacteria sticking together and forming biofilms. We also showed that when we block this antigen, we break up bacterial biofilms. Using this knowledge, we developed a breakthrough antibody technology that inhibits uropathogenic E. coli biofilms. This technology can prevent biofilm formation at nanomolar concentrations, dismantles existing biofilms, and can enhance the activity of antibiotics that are not effective against biofilm infections. The competitive advantages of our technology include broad clinical applicability. We are addressing 80% of UTIs. This technology aligns with the WHO innovation criteria for new anti-infectives. It is expected that this technology will lead or will not enhance resistance development because we are targeting virulence, not viability. And the technology has therapeutic flexibility as it can be used as a standalone therapeutic or in combination with antibiotics. Therefore, our technology addresses both clinical efficacy and commercial viability. Our founding team includes experts in protein chemistry, biophysics, biofilms, and UTIs from Trobe University and also the University of Queensland. Our advisory team has experts in antibody technology, clinical translation, and commercialization. We have also strong support from our patient partners, which come from different patient advocacy groups, including Chronic UTI Australia and Live UTI Free. We recently secured $1.5 million from the Australian Economic Accelerator Program and from private investment to take this technology to preclinical stage. And we also have a strong intellectual property protection, which covers us internationally. We think we have a technology that can change how we treat urinary tract infections, can rescue antibiotics that are not efficient against biofilm infections, and can reduce the development of resistance. Thank you.
Thank you, Yogna, for this. It was a very good but a very important one to sort of show that connection of what is actually happening with the basic research into the actual innovation. Also, I should say congratulations to Begonia, also to Jason for their ARC DP success yesterday. Won a very competitive round, got some very nice news and sort of be able to help continue celebrate afterwards. So now, while we set up the panel stage, I'll actually ask the crowd to start getting your questions ready. We'll invite our speakers to come up and sit with us and start to get your questions going. And then hopefully we still have our Boston contingent will be awake and joining us. Thank you. So let's give a warm welcome to all our speakers joining us on stage once again, and thank you for their talks. And I'll start with the first question to help get things going. So, Richard, we have researchers working on early stage projects in the antimicrobial resistance space. We are currently preparing work plans for early stage grants and non-dilutive funding schemes with longer term goals of applying for CARB-X funding. Could you advise on the types of data and evidence? You touched on it before, but what are some particular things you look for with the data and evidence that they will need to be that really competitive in those CARB-X funding applications?
Yeah, I think that's a question we often get. I think because we fund at different stages, we fund in stages, and so a program can come into the CARB-X portfolio if it's within scope of the funding round at any stage between what is hit to lead and the first time the molecule is evaluated in humans. And for hit to lead, it doesn't need to have, a lot of people say, well, does that mean you have to show efficacy in an animal model? No, that's not true. We will take programs before that stage, What we do require for, if it's a direct acting molecule, is we do require activity, wholesale activity against a wild type pathogen that's within the scope of the call. So not an efflux mutant, but a wild type pathogen. We'd like to know what the target is. We have taken programs where the target is unknown if the hit has come from a wholesale screen, say, and we will work with the company to set up an experimental plan to try and determine that target. But quite often we like to know the target. I think the key is how differentiated is the product going to be, and that's what in the application, that's what you need to highlight. And think about what your end product will look like and how that will bring value. And so getting early pharmacokinetic data, whether there's a potential for oral delivery as well as, say, IV, things like that will differentiate the patient populations you can treat and will increase the flexibility of your preclinical work to try and maximize those differentiating characteristics that your product may bring.
Thank you, that's great. Are there any questions from the audience?
Hey Richard, I'll ask you another question. Thanks for coming out before you fall asleep. I'm just interested, like with pharma companies, they're used to antibiotics, they're used to vaccines, but you know, we got biologics coming through. What's, you know, are pharma companies keen to pick up on sort of new technology, sort of such as like biologics, you know, like peptides and proteins and things like that, or are they still stuck on antibiotics and vaccines?
Different companies. Well, first of all, congratulations, Jason, and also to Begonia for your grant yesterday. I did see that announcement, and I think that's fantastic. So congratulations. Thank you. I think there are certainly companies that are willing to take more risks, and that's really embedded within the culture of the individual company. I think if you take one of the largest companies that are still actively in the discovery space, like say Roche, they're focused on a pathogen and an indication rather than the modality. So they say, you know, for example, they may be really interested in, say, Pseudomonas and lung infections. And so as long as you can provide data to show that you are targeting that pathogen in that indication, the specific modality is less of a concern to them. But what they will require will be evidence that you understand your molecule, how it will be used, and will it bring value either as a standalone product or in combination. I think that more and more companies, I think, you know, in terms of your vaccines, I think while some companies are still focused obviously on vaccines, there's more and more companies that are looking at other preventative modalities. And I mentioned some of those. You know, we obviously support decolonization through, say, live biotherapeutic products or microbiome modifying products. Monoclonal antibodies, especially with the Gates are now funding a large challenge to try and reduce the cost of monoclonal antibodies. So I think that in certain populations and certain indications, the use of monoclonal antibodies as a short-term preventative measure will be extremely valuable, especially if the price can come down and open up the markets into lower resource settings. So I think companies are looking at the different, I'll say, non-traditional technologies. I think what it takes, as you know, Felicia mentioned with the mRNA technology, it's that right time, right place to show clinical evidence that it's going to be useful. And I think, you know, Moderna did that beautifully when COVID struck. But I think what a lot of companies are now looking for is some clinical evidence that some of these non-traditional approaches can actually provide some clinical benefit. And I think that will then explode the field open.
Thank you.
Thank you. Any other questions? Oh.
Felicia, I'm wondering if Donald Trump's antipathy to messenger RNA technology Robert Kennedy's antipathy to vaccines in general, is that going to make any difference to what Moderna plans to achieve? And particularly, will it make any difference to what the Australian arm of Moderna will be doing?
Sure, it's a great question, and I was expecting that we'd probably have something like that come up. So, look, it's really interesting. So it's during the first term of Donald Trump's administration that there was mRNA technology really came to the forefront as part of COVID-19. I think, and it's public knowledge, we had a contract with BARDA for sort of pandemic influenza, which got cancelled. The truth of the matter is Moderna is still committed to develop what it needs to develop to support the globe. And I think really there's an opportunity, and what we're seeing is really the resilience markets, so the UK, Canada and Australia. The support is there, and the belief in vaccines, and I must say the belief in the technology as a platform beyond vaccines. So, as I mentioned, of course, we have the respiratory portfolio, but we're seeing an uplift really in oncology. And I think that when we see products coming out in the oncological space and that value, you hope that there will be a change to some of the rhetoric. I think another thing that's really interesting, so we were projecting that we'd get a reduced rate of vaccinations in the United States. We actually exceeded our expectations. So whilst the rhetoric out of the government is a particular way, I think people are making their own choices for the protection of their health. Yeah.
Follow-up question for you, Felicia. You've got a very impressive R&D portfolio. Can you share some of the roadblocks in getting your products to market? I know the investment with the COVID-19. What
Sure.
specifically is stopping some of those other projects that's actually going?
So I think the thing is, when you have a platform that is so sort of capable of being changed and putting in it's picking what next, what do you do? And look, the priority for us at the moment is the respiratory portfolio, because that's where we'll be able to generate income that will enable us to then feed more funds back into the other assets and targets that we have there. I mean, and we look at our success rates from transitioning from phase 1 to phase 2 and phase 3. So if you look at industry standards, we're actually significantly higher travelling through those, because we're gaining information on the platform each time we do a clinical trial. And so we're not seeing the same level of failure rates that you see with traditional methodologies around phase 1, phase 2, phase 3. And so you really have to pick, well, what's the one that makes the most sense to push forward? And so that's sort of a real concerted effort from our leadership to, what's right for the markets that they're looking to tackle. And I think, having the broad capability, particularly in some of the, the rare oncology, respiratory and infectious diseases, it comes down to, funders like, so the World Health Organization and CEPI and, Wellcome Trust that might be interested in that infectious diseases space.
Hi, Felicia. Sorry, it's you again. My question really is more around the regulatory environment and how things have really progressed. And it's great to see Moderna and BioNTech moving through new technologies, increasing their portfolios, new assets coming through. But as we know, that the regulatory environment took some way, at least after the COVID, to sort of adjust with this platform approach. Mm-hmm. And I'm just wondering if you can comment on how things have gone there. And how that sort of impacts sort of a pandemic preparedness for future.
Sure. I think, look, you've got to, realise they're learning as we're learning, and so we're in a journey together. And I must say, the TGA. So that's not my area. My colleague Carolyn Chuchek-Szabo deals with the regulatory side, and I know that there are really great, strong relationships that have been formed, and all decisions that they're making are evidence based. Which is critical for acceptance. I think there's a lot of work that can be done across, harmonisation globally when it comes to regulation. But I think those regulators are really open to conversations, particularly around platform technologies and the capabilities. So the more data that we generate, the more applications across a broad portfolio, people start to become comfortable. So you see the similar sort of issues with CAR T therapies, those sorts of things. So different platforms require different regulatory approaches, and I think there is an openness to learn, to innovate the regulatory science, and there's a lot of work being done in that space.
I'll ask you, Marilyn, you've been straddling academia and industry. What advice would you give to people?
Oh, that's an interesting one. Well, first of all, one has to know how to set up a company and how to deal with regulators and be realistic before you start. So I think when we started, we had a blue sky approach that we would save the world, but I find going through the regulators has been the hardest thing. And also, you need to know how to raise money. Because the further you get along the track, the more and more and more expensive it gets and the more difficult.
Oh, that's great.
Thanks. That was excellent. I actually have a question for Marilyn and Begonia. I think this might be relevant for the pair of you. With regards to where your therapies are operating, how do you see yourself fitting into the clinical treatment of your respective target illnesses? And do you feel concerned about, in a way, almost being too good and sort of being put on the shelf in that reserve category of therapeutics where perhaps you might not get the same kind of return that potential companies might be expecting from your respective therapies? Are you too good at what you're doing?
In terms of our technology, one of the advantages of our technology is that it's not killing bacteria. It's not... So basically, the expectation is that it will not lead to resistance development, and in fact it's helping antibiotics work against biofilm infections. So it shouldn't be a technology that would be put in the shelf. It should be a technology that could be used as on itself, or together with antibiotics that have become obsolete for therapeutic treatment. So
2 minutes, okay. Look, I think it depends what you're trying to do. So if you come up with the new world's best antibiotic, the worry has always been that it'll be put on the shelf for when there's a real emergency and it's really needed. So they'll try everything else first and they'll use that as a last resort because it needs to be protected. And that's been one of the reasons why the big farmers haven't put a lot of money into developing new antibiotics. In our case, the products that are out there don't work at all. So, I mean, they do work. Imagine after spending $8,000, 1 whole year of treatment every day, and you've only got a 17% chance of a cure. So I think that it's also, what the market wants.
Thank you. For Marilyn and Begonia, I just wanted to get a sense of what scaling might look like for you both. Are you looking to stay on shore or likely go offshore?
I'll answer first this time.
Yep.
Okay, so with Hexama, with a board, a very senior board, they thought it was best to go offshore and do all work in the United States, not the work. The innovation happens here, but commercialisation in the US, because that's where the big market was, US and Japan. However, today I've been talking to this new scheme, industry growth scheme run by the Australian government, and they want us to try and do our best to develop products onshore. So with our new little company, we are probably going to try to start to work in Australia, but hoping that it gets big enough to go international. Once you want to work with the FDA, it costs a fortune. We spend a lot of money trying to satisfy the FDA requirements. We've done a lot of that. We hope that we don't have to do it again here in Australia with the TGA, but we will have to do more.
I guess our vision was to follow FDA indications, FDA regulations, because if we get approval in that jurisdiction, that would apply to others. But it is expensive, and so yeah, that's our initial thought. But I don't know about that.
Well, the final thing was to fund FDA. You really need venture capitalist money. That's normally in the US, and normally those investors in the US like the work to be done in the US and don't really trust work that's done in Australia. That's what they told us.
I'll do one final quick question for tonight for Begonia. So you've had a very successful 2025 and had all the grants and all the other success. What's your hope for 2026?
Academia is a roller coaster, so I don't even want to say anything, but just maintaining. I guess Jason and I, what we are trying to get is further funding to be able to support the technology to the next stage. So that's what we are trying to do. We have a few grants and fellowships on the go as well. So yeah, just look, to stay as we are, that would be perfect.
Thank you. And thank you to all our panellists for sharing such valuable insights and expertise. We deeply appreciate the time and knowledge and the investment that you've made in this event and to make this interesting, illuminating and inspiring for all. So would you all join me in giving it another round of applause, and thank you to everyone. And in closing, I would like to sincerely thank all the support in attending this La Trobe Industry Innovation Series event. I'd like to actually also acknowledge Hope and her team as well, who always make it look easy to put on a difficult event like this and make things run seamlessly. So through the industry-university collaborations, we are actively bridging the gap between academic innovation and commercial application, turning groundbreaking research into real-world solutions and improve lives and shape our future. This is what our slogan for the university is: the impact is real. And I hope you saw that through all the talks and all what's happening. This is how we see ourselves and see what our impact is out there in the world and what is actually occurring. So with that, I'd like to invite you all to stay, connect and enjoy some great company food and refreshments. We'll be here till 7:30. You have heard all the different stories from the different people. Make sure you ask them and go further, talk to all the different people around and make new connections. Thank you.